morning. Oh, the microphone's on. Good morning, everyone. I see coffee. No one got me any. I feel like I saw you guys just a couple of hours ago, maybe dancing. Good moves over on the dance floor. What a wonderful night. What a wonderful couple of days this has been. Is everybody good? Happy, enthusiastic? Yes. Thank you. I know we are smart people here, the doctors that are on hand, and I believe we are absolutely doing the right thing to have this primarily virtual for the 2021 Cancer Symposium. But thank you for being here and masking up and being socially distant, and thanks to all of you watching virtually as well. This is always such an important part of what we do here every year at Napa, and it is going to be just as good, and I think just as thorough. We're all going to learn so much about finding the ways to beat cancer through immunotherapy. So I wanted to just thank everybody for making it happen virtually to make sure everybody's as safe as humanly possible. And that's what we did last year, right? So this is progress. So think back to last year where none of us were seeing eyeballs. So this is awesome to see all of you in here, and at least we're all here on property. Welcome officially again to the 2021 Cancer Symposium. And I am thrilled, and I thank those of you who were at the dinner the other night. I mean it when I say I'm so excited to continue to learn more about how we're battling this disease, and immunotherapy is such a key part of that. So we're going to have 4 awesome doctors come up and talk to us about that today, give us their presentations. We got to thank our sponsors, because the sponsors have absolutely been incredible and always are. And this is not just because I work for ESPN and love my job, but I was told this is the first sponsor I need to mention. The V Foundation's founding partner for 28 years, almost 30 years, has supported the foundation through 2 primary fundraisers, and you all know what they are. SB Week in July and V Week in December, and I think that's going to be here before we know it, right? For more on that, and to talk about why today's symposium is so important to ESPN, I'd like to welcome a very good friend of mine, but I got to read his official title because he's kind of amazing. ESPN Vice President of Corporate Citizenship, Kevin Martinez. Come on up.
Thank you, Sage. So first of all, you guys see me come up every year, and I was telling my dear friend Rita Farrell that this is the time that I don't get to talk about numbers and sports, and I get to bring out a piece of me that is so important, which is the conversation around cancer and the support that we have of 5,000 employees at ESPN that believe in you in this room like no other. I guarantee you we are your greatest champions. We know what it is to do this. As I always say, I will never be the smartest person in the room, but I will be sitting by them. So the good news is I can sit anywhere in this room and feel really good because of the work and the research that's being done. So I wanted to start that way first. Second of all, I just wanted to blame a couple people for what you're seeing today. So Jane, I blame you. Shane, I blame you. Amanda, Shasha, Dr. Nelson, I blame you for this because I had the greatest night last night. I really did. The beauty of this is to have people together to believe in something, a shared value, and to come out and also feel like you're part of the family. So I won't take my hat off for you because that'll be scary. But I blame you for this, but I blame you in the best possible way that you are the most amazing hosts, and you really are doing amazing stuff. And I also just want to put a real quick plug in that when you see them, please do thank them, because what you know and don't know is that this has changed about 20 times. And to be able to do this, we know this. We just did the NBA Finals in a bubble and, wide world of sports at Disney, and, the combination of planning and progress, technology, it's hard. And these are amazing, best in business, special event people. So could not be more proud to do that. Second of all, I want to thank Blackbaud, who is an amazing organization that believes in the soul of doing right and doing well. So Blackbaud, thank you for your continued efforts. It's phenomenal. And then I also now want to drill into something why it's really personal to me. I lost my mom to COVID this year. If I cry, I apologize. But my mother-in-law had breast cancer about 20 years ago, and she is a survivor. But in the chemotherapy, it damaged her heart. And she is an angel on earth. She's in Boise, Idaho, and she's getting immunological treatment. She has esophageal cancer. It's come back in tumor form. And again, I'm dangerous because I can't speak to what the scientists are. But her tumors are shrinking. They're getting smaller. And she unfortunately fell down in a grocery store yesterday because she wouldn't use her walker. But she's out and walking and doing great things. But that's because of you and the science that you have put together. Now we've got to figure out, I heard someone say that, sometimes cancer causes little roadblocks in the front when you get to immunological. The heart piece, she has to be pulled off this particular immunological cancer drug, but we know that there's a vision and there's a future out there, and that's because of you. So I just want to thank you all so very much for all that you do. Thank you very much.
I love Kevin because he's amazing at his job, and I've known him for, gosh, the entire time I've been at ESPN. But I learned something more about you today, and I thought I knew a lot. I know he drinks really good red wine, and I know that if someone comes to my house during a party and spills red wine on my white velvet couch, I know who to call to help clean my pretty white couch, right? Kevin Martinez. The stain is almost gone after 3 years, and I keep scrubbing. But it wasn't, we know who it was. Everybody has a personal tie. And that's the heartbreaking thing, right? But I think that's the beauty of what brings us all together here and continuing to battle and to fight. And I think it's actually just such a great lesson in life these days, right? Especially these days. But you can look around and it looks like everybody, they've got all it together and their life looks so perfect and you don't know what people are dealing with. So thank you for sharing that. And I think vulnerability is such a beautiful thing. And when you are able to speak a little bit, and I think Jay and I will share a little bit more tonight as well, but you realize that you're not alone. And we know we're not alone today with these doctors here who are going to help us feel a little bit better about what has continued to happen research-wise, despite a global pandemic. So thanks to ESPN. Thanks again, to Blackbaud, the world's leading cloud software company powering social good. We are nothing without our sponsors, so to both sponsors, one more time, thank you. Okay, again, time for the smart people. No offense, I mean, you're smart too. But 4 doctors at the top of their fields, and we learned it the other night, and you know them. Their passion is incredible because they will not give up until they find a cure. And the immunotherapy part is just brilliant to me, and there's still so much left to learn. So here we go. Again, 4 doctors. Each doctor has 10 minutes. Starting with Nelson. I will pull you off the stage if you don't stick to 10 minutes. I'm kidding. I love him. I could listen to him talk all day. But after each presentation, we're going to take some question and answer as well. Well, we'll take the questions and try to answer. Isn't that how Q&A goes? So we'll do that as well because I think throughout, questions come up, and we'll get them on the board and share them with you. And then if anyone out here has questions as well, we'll do our best to get them in. So here we go. He's the host chair of the Scientific Advisory Committee, director of the Sidney Kimmel Comprehensive Cancer Center. He is one heck of a guitar player as well. I love him, Dr. Bill Nelson. Come on up.
Thank you, Sage. I thought what I would talk a little bit about today, it's impossible to talk about cancer, healthcare, or anything without talking about SARS-CoV-2, COVID-19. And the last 18 months, this virus, and that structure of the virus has practically become a television news logo, right? I mean, you see this all the time. But it's had a dramatic effect on cancer care and cancer research. Cancer patients are somewhat more vulnerable to get infected. They get a little bit sicker when they get infected. They're harder to vaccinate. So this is a significant burden on cancer care as well as all of healthcare. Our researchers confronted bans on going to work, bans on traveling. So clinical research came to a halt and then was restarted. Laboratory research to a halt and restarted. The V Foundation aided the V scholars, aided these translational researchers through this process by giving them a little bit of room, and I'm happy to report at this point, despite Delta variants and the like, cancer research is up and going again. We're starting to make progress again. It's starting to work again. But look, what is it, 200 million cases worldwide, 4 million deaths. more than 30 million cases in the US, 600,000 deaths. What's going on? And there's some good news to this related to the investment in cancer research, because it's paying off in how we're responding to COVID-19. If you look at that structure more carefully, focus on the S protein, the red dots coming out. That's the spike protein you hear so much about. It forms what looks like a corona around the virus, hence the name of these viruses. And it's a cousin of viruses seen before. Huge debate on where exactly it came from, which we're not going to get into today. From the perspective like we think about cancer, what are the targets that we can chase after to build treatments, to build vaccines and the like? Two jumped right out. Now remember, the first recognized case that's confirmed, December 1st, 2019 in Wuhan, December 31st, declared a pandemic by the World Health Organization, first cases in the US in late January. Quickly, because of all the technology that we've come into in modern medicine and cancer research and others, the virus is sequenced. Tests are built. The people who do these tests, molecular pathologists, are the same people that sequence cancer genomes. So that asset turned out to be incredibly valuable, the PCR test. So here's what we learned quickly. There's one target, a protease. That's a protein that cuts another protein into two. It helps the virus enter the cell. The way the virus sticks to the cell and the way it gets clips up to enter the cell is why these variants are more infectious. They stick to the cell better, they climb into the cell better. That explains the Delta variant. One way to look at it is that common flu, one person typically infects two, right? The original variant of this, probably one person infected three. So one infects three, 3 to 9, 9 to 27. The Delta variant is probably up in the range of 5 to 8. So one infects 8, 8 to 64, so you can see how it explodes very quickly. Chickenpox, just a little bit more. World indoor record, just for sake of argument, is measles, which is 16 to 18 or something. The key to a protease is that it's like Arnold Schwarzenegger in that old movie, you know, if it bleeds, we can kill it. If it's a protease, we can inhibit it. Okay, so there are going to be drugs that stop proteases. HIV protease, that's why we can do so well against HIV. So this is a great target. And the other is this spike protein. This is what you respond to when you recover, the people who recover. So you can take their antibodies early on. They tried to use that to treat people, and we can make antibodies to this. The thing we first did in cancer, we made antibodies to Herceptin. This is the kind of thing we can chase after. So what treatments have we approved? Remdesivir was somewhat on the shelf, a nucleoside analog of viral genome replication. This will get better, I suspect, with oral medications and the like. Antibody drugs. This technology was incredibly influenced by investments in cancer research. It largely arose out of a cancer research logic. Combinations of these things, you can see the spike protein, they stick to more than one spot. That means if there's a variant so that one antibody doesn't stick, chances are the other one will still stick. I was a soccer player, you kick them in the knee and the ankle at the same time and they will fall over. Immunomodulatory agents. This is a kind of a boring drug. It's a steroid drug. If you slow down the immune system, the response to some people, the immune response is overwhelming. It's what makes them sick. Some others, it's the virus that overwhelms them, so you have to take a look at that a little bit. Another drug that affects the way the immune system works, keeps people in the game till they can clear the virus, baricitinib. And of course, we're in the middle of now trying to deploy these vaccines. Think of how quickly we came up with these vaccines. Operation Warp Speed, I don't care who gets credit for it. It's incredible. BioNTech, Pfizer, Moderna, the J&J, you know all about these because you hear about them on the news. What you may not know as much about is this technology of using mRNA. They're the blueprints, the working copy for all the components of cells and tissues, the components of the virus. This was used in an attempt to build anti-cancer vaccines. You're going to hear a little bit more about those later today. But look at all of these things. As of 2017, we never heard of SARS-CoV-2, never heard of COVID-19. All these were clinical trials trying to use mRNA to be parts of vaccines for cancer. All the stuff, the trial and error in this case, how to make the RNA so it doesn't get eaten up in the middle of the cell, all these kind of things were what was going on. So we were ready, because of the investments in cancer research, to take on COVID-19 at warp speed. Today we're going to hear about immunotherapy of cancer. This is the idea that the body's own immune system could be harnessed to seek out, attack, and destroy cancer cells is not a new idea. And there are people who've dedicated their whole lives to it. You're going to hear from some of them today, right? But there were immunotherapies at the beginning. This witch's brew from this character, William Coley, was a mixture of bacteria that were killed. This is insane. They would give these people the stuff, they get incredibly sick, shake like a leaf on a tree, etc. And some of them seem to do better, to the point where Park Davis actually sold this as an over-the-counter preparation. Boy, we've gotten better. The scientific question that we've been managing in the V Foundation through applications, V Scholars, was if the immune system can recognize my cells and your cells differently, so much so that if you transplant your kidney into me, my immune system will reject it. If it can be that careful in discrimination, if it can tell a bacteria or a fungus or something from your own cells, right? Can it tell a cancer cell from a normal cell? That was one of the great questions, and the answer is, of course, that it can. That means that we can build cancer immunotherapies. Here's cancer cells growing on a dish. Here's some immune cells. They're checking this guy out, and they know that this cell is not a normal cell. The secret now is, if they can recognize it, can we endow them with an attitude so that they go out and seek and destroy the cancer cells? The cancer cell's going to fight back. It's going to be tricky. It's going to figure out a way to hold them at bay, to sneak around them, to eat all the food so they don't have any left to eat. All these kind of things are what our researchers are working on. And so you see this long timeline, but the last few years are when things have exploded. New approvals for immune checkpoint inhibitors. This is changing the game entirely for lung cancer treatment, melanoma treatment. So-called CAR T cells take the immune cells, endow them with an attitude, put them back in the body, let them chase down cancer cells. Changing the game for certain leukemias, myeloma and stuff. This is growing, and there's no turning back. This is really starting to work, and eventually it's going to get us to victory over cancer. My favorite thing in the Jimmy V speech has always been when he starts to act like a coach in the middle of that very emotional talk. How do you go from where you are to where you want to be? And that's the challenge for us when we challenge our young researchers to take things on. You have to have an enthusiasm for life, a dream, a goal, and you have to be willing to work from it. You're going to hear people today that have that enthusiasm, they have a dream, they have a goal, they're definitely willing to work for it, and they're going to share with you what they can describe for you today. So thanks.
I could listen to him talk all day, and now I know if I say anything wrong, he'll kick me in the knee and the ankle at the same time, and I'm out, right? Thank you so much, Dr. Nelson. Let's keep it going. She is University of Pittsburgh Distinguished Professor of Immunology and Surgery and the founding Chair Emeritus of the Department of Immunotherapy. So the perfect person to continue this discussion as she gets the microphone in her hand, Dr. Oya Finn. Hello again. Come on up.
Thank you very much. Thank you for all of you who are here, because I'm sort of an actress. I need the audience when I talk. And I want to tell all of you who are watching online, I really miss you here. But we'll catch up tonight. And if you have any other questions after meet me at the marketplace and we'll discuss it. So I will give you an illustration by using a personal example that I say, I call traveling the cancer vaccine road and reaching my destination. It's basically, where you are, where you want to be, and how you get there. And it's been a very long journey, which I'm very happy to share with you, because I'm really proud of it. I'm coming sort of to the end of that journey, hoping to make other people enthusiastic about what I do so that they can carry on. So I started the journey in cancer research as a graduate student at Stanford between 75 and 79, and also as a postdoctoral fellow at Stanford from 79 to 82. Both of my mentors were passionate about solving the cancer problem. My graduate program mentor, Henry Kaplan, was a radiation oncologist, and he is responsible actually for solving the Hodgkin's disease therapy question. He's credited for creating the treatment for Hodgkin's disease. And then my postdoctoral mentor, Ron Levy, was very interested in whether or not, as Bill questioned, whether the immune system can actually see cancer, and he is a medical oncologist working in B-cell lymphoma. So I was indoctrinated very early on. I was a graduate student in microbiology and medical microbiology, actually, that had a concentration in immunology, and I really wanted to, influenced by these two incredible scientists, to follow their passion and figure out if we can use new knowledge about the immune system and whether or not it can see cancer. So I was able to start on that on my own when I joined Duke University as an assistant professor in 1982. But as I mentioned the other night during the dinner with the docs, I was advised very strongly not to start a laboratory in cancer immunology because, as I mentioned the other night, it could be a career suicide. So I gave it a very sophisticated name, Laboratory of Cellular and Molecular Immunobiology. So in secret, with a nice group of young students and technicians, we were in secret looking at the immune system and cancer. And we sort of took the privilege, and I'm actually showing this slide more for Bob Bast, to remember what an idyllic place this was in the early 80s, because Bob was my cancer center director at the time. And we wanted to use the privilege of being able to ask questions and being supported to do that, to ask this very important question and not reveal what we were doing until we actually had the answer. The other thing that was very important was that at that time, just around the middle 80s, science was really, immunology was really progressing as a science. We were learning some very basic facts. And one of the things that was very encouraging is we learned how to grow human immune cells in culture. And the growth factor for these, for these cultures was discovered. It was interleukin-2. And so we were encouraged by that and asked the question, if you take a piece of tumor and put it in culture and a lot of immune cells migrate out, could we ask these cells, what is it that they see? Why are they there? Do they see something in that tumor that, and simultaneously we were looking at, as Bill mentioned, at kidneys that are being rejected, and the same thing was happening. T cells were coming out, immune cells were coming out of little biopsies of rejecting kidneys. So we thought, if the kidney can be rejected and these cells are there, perhaps these cells are also in the tumor trying to reject it. And can we ask? And without giving you the details, I can tell you that in a very short amount of time, isolating these cells, being able to grow them, we found out that, in fact, they were there recognizing the tumor, at least some of them were recognizing the tumor, and in fact, we were able to find out why and how they recognized some of the molecules that are expressed on the tumors differently than normal cells. So on the left here, right there, you see a normal pancreas, and in brown, you see the molecule that I will continue to talk about, mucin-1, MUC1. And this molecule is expressed on the inside of the pancreatic duct in very little amounts and in a very specific form, so we call it fully glycosylated form, that protects that duct from the harsh environment of the pancreatic secretions. Now, when one of those ductal cells becomes transformed and grows into pancreatic cancer, this particular molecule changes tremendously. You can see all that brown. First of all, the ducts are full of tumor cells, and the molecule is expressed at a very high level, which is the picture on the lower. And the immune system recognizes both the amount of the molecule that is expressed, that it's more than it should be, and that the kind of a molecule it is, and the molecule was losing its sugars and revealing its structure, its protein, its backbone to the immune system that had never seen it before on normal cells. So we hung on to this discovery and decided that this was, and in fact, this was the very first tumor antigen without any mutations or anything. It was expressed differently on tumors than in normal cells, and the immune system was reacting against it. Not only did we find T cells against this molecule, but also in sera of these patients, we could find antibodies that recognized the abnormal form and not the normal form of the molecule. And the minute you do that, you begin to dream about vaccines. If you have a target, let's target it. And the way we knew at the time to target things, it was stemmed from what we knew about infectious diseases. You target viruses based on their molecules, like the spike protein on the coronavirus. So we decided to work on vaccines. We published these two papers in 1989 and in 1991, and I want you to see that in the 1991 paper has actually Bob Bast on it. I bet you, Bob, you had forgotten about this paper. But Bob was instrumental in providing me with the tools, which was samples from patients, to actually show that these cells that we were isolating were killing patients' tumors in tissue culture. And then hopefully, so the whole idea was, let's make a lot more of these cells in a cancer patient. Perhaps they don't have enough. Let's vaccinate them. Let's boost those cells. Let them go to the tumor and destroy it. So this was such a big discovery because it was the very first human tumor antigen actually to be discovered as a target of human T cells, that Pittsburgh came after me, and the Pittsburgh Cancer Center director at the time, Ron Herberman, wanted to start a cancer immunology program, and he wanted me to start it and lead it. And so I left this idyllic Duke environment, but I thought about it very hard. I don't know if you can read this, as I wonder where we go when we die. Pittsburgh? You mean if we're good or if we're bad? So I was trying to figure out why they were getting me. I figured because I was good. So I decided I should go. And in Pittsburgh, within 2 years, because we started the first in the world clinical trial of a cancer vaccine based on your own molecule, just differently expressed in a different form, no mutations, and we started this clinical trial in patients with breast, colon, and pancreatic cancer. And we were on the front page of the Pittsburgh Post-Gazette, the main paper. We were on all television stations, et cetera. And this was the beginning of the dream that we can create a vaccine as the immunotherapy for cancer, that to either generate an immune response against this molecule that people don't have, or boost the immune response that they're trying to make, but it's not strong enough. And one thing I should mention is that this molecule is expressed on over 80% of all human cancers, because they're all derived from the epithelial cells, ductal epithelial cells. They're all adenocarcinomas. So it's breast, colon, pancreas, lung. They all express this molecule in an abnormal form that can be recognized by the immune system. So between 1993, when this first trial started, and 2008 or 2012, when the last trial finished, we ran this trial of therapeutic cancer vaccines. And the entire goal of these trials was to show to the FDA that these vaccines are safe. The problem was that, as Bill mentioned, often the immune system for a cancer patient is compromised by the cancer, certainly by the treatments, so that the vaccines don't have as good a chance of generating a very strong response as we would like them to have. And while we were doing these trials and trying to figure out why the responses we were getting was low, didn't really affect significantly the clinical outcome of the patients, the whole field of tumor microenvironment was developing, learning how patients' immune responses are controlled by the tumor and the various other factors that the tumor encourages to be made. And most of them, and you can see, the tumor, if you imagine it, is not a ball of tumor cells. There's very few tumor cells here. The tumor is a ball of all sorts of cells surrounding the tumor cells, and many of them are very unfriendly to the immune system. So we learned from basically mediocre results in the setting of therapeutic cancer vaccines that the tumor was trying very hard to suppress the immune responses of cancer patients and not allow them to respond to the vaccine that well. So having learned that, and as I said, benefiting from other people's work on the tumor microenvironment, and even contributing to it by our results from our cancer patients, vaccinated patients, we remembered that, in fact, I showed you the front page of the Pittsburgh Post-Gazette, but inside, we had been interviewed by people, and my colleague Mike Lotz, who ran the trial, and I stated that this is really just the beginning, but someday we hope to inject people at risk for developing cancer. And those of you who were at dinner the other night remember me saying, I would love to prevent the diagnosis of cancer, not just to treat cancer, but to prevent cancer from ever being diagnosed. So we were first then, in 1993, trying the first therapeutic cancer vaccine based on a tumor antigen that is not a virus and not a bacterium, but your own molecule. And we decided to be first again, take the bull by the horn. If we wanted to do this, let's do it. And with my colleague, Rocky Schoen, who is a head of our GI division in Pittsburgh, we decided to test it in a preventative setting. The same vaccine that we were testing in cancer patients under different conditions in the preventative setting. So we decided that to look at colon cancer, that's Rocky's interest, and we knew, and we could determine, that abnormal expression of MAC1, of our target antigen, starts with the growth of a polyp. So it's normal on normal colons, but a small adenoma, large adenoma, begin to express very high levels of this abnormal molecule. Of course, they're much higher in cancer and much higher in metastatic disease. And we were doing vaccines right there. So all we decided to do is not to change the vaccine, but to change the patient. So now we wanted to do the vaccine right here. There's a polyp diagnosed, you take it out. You don't want people to get more polyps because that's a higher risk for developing cancer. So can we prevent the polyp from recurring just because the polyp will show that same antigen that you immunize against and that the cancer would have? So we did that. Here's the culprit. This is removed. The important part is that people with this advanced polyp, in 1 to 3 years, 48% of these patients would recur or come up with new polyps, and each one of them is at risk for developing into a stage 1 colon cancer or later, depending when the colonoscopy is done. So we decided, we started a trial. We convinced the FDA that at the level of our institution, we can safely run this trial in patients with a history of these polyps. We vaccinated these patients after the polyps were removed, and all we wanted to know is, can you induce an immune response in these patients? How does it compare to cancer patients? And is it safe? And we published a paper in 2013 showing that, in fact, the vaccine was very safe. We had absolutely no side effects of the vaccine. And we showed that over 50% of our patients developed very good antibodies and T cells specific for this vaccine. This compared to 2 or 3% where we could measure that immune response in cancer patients, regularly advanced cancer patients. And this dotted line that you may see here, right here, is the level of the response that we were able to get in cancer patients. Our healthy individuals at risk for cancer, for colon cancer, had 40 to 50-fold higher responses than we ever saw. So it told us that the vaccine was good. It was just at a very bad setting if you do it in a very advanced disease. And more importantly, it induced a memory response. So the first 2 or so injections generated the immune response. We're looking at antibody. It started to go down. When we boosted it in 1 year, it went back up. And this is what you want. You want to induce an immune response at the moment to control, to expand, et cetera, and to go into a memory response. And then you have protected the person for long term. So with this data, we could go to the National Cancer Institute Division of Cancer Prevention and say, now we want to know, does it work? We know that it's immunogenic. The vaccine generates an immune response. We know it's safe. Does it protect from polyp recurrence? And we were able to do 7 medical center trials under the funding from the Division of Cancer Prevention through a cancer prevention network that they have, and we asked the same question. Safety, is it immunogenic? But now we had 110 people, 55 placebo, 55 vaccinated. Does it make a difference in terms of the outcome of these patients in terms of polyp recurrence? And the study was such that we had some power to actually see if there's a difference. Here's the result. We just unblinded it recently because it was a double-blind trial. And the blue people are vaccinated. The red are placebo controls. You can see, just like in the first study, we had some of the non-responders. We know why they're not responding. I don't have the time to discuss that, but I can do that offline. However, the important thing was that we can determine that the placebo controls, right here, 66% of people at year 5 had new polyps. Vaccine non-responders, those that did not respond to our vaccine, 63.9%. But those who responded to the vaccine, 27% only grew polyps. So this is the very first time, really ever, that we could test an efficacy of what was a therapeutic cancer vaccine used in the preventative setting. And actually, I'm coming to the end stage. It took over 3 decades to come to this point, to open the door to preventative cancer vaccines. Other than HPV, we have not been doing that. I think we should be doing a lot more of that. It took 3 decades of basic research in my lab, and these are 3 decades, smiley faces from 3 decades of my lab. And it took, just for the last trial, the 110 people trial, efficacy trial, it took this whole trial team involving the NCI Division of Cancer Prevention, the Cancer Prevention Network at Mayo, my laboratory, 7 study sites, all over the place, including Puerto Rico, and the company that provided the adjuvant for it. So it takes a lot of people, a lot of money, a lot of time to actually realize a dream, and I really think this, this is opening the doors for the future use of cancer vaccines in prevention. And just the last slide is MUC1 is one antigen. Here it is, and we have, these are the premalignant lesions that are positive for this antigen could be targeted. So pancreatic intraepithelial neoplasia, that's how pancreatic cancer starts in many. You can perhaps stop it. Barrett's esophagus, then polyps like I showed you. But look at all the other antigens that we have been using in therapeutic cancer vaccines that can now be used in the premalignant setting. These are all diagnosable premalignancies where now you just survey people or surgically remove it. Perhaps all of those can be targeted in addition to other treatments or in lieu of other treatments with preventative cancer vaccines. So I am hoping that my 30 or some years of work with this antigen has given an example that many other immunotherapies will follow in the future. Thank you very much.
And thank you, Dr. Finn. I mean, what if you had listened to all those people 30 plus years ago who said it's career suicide to try it? What if? Never listen. Thank you. The amazingness continues now with the Vice President of Research and Development, and also she's an adjunct professor of microbiology and immunology at the University of California, San Francisco. Dr. Lisa Butterfield did not have to drive very far, right? You're kind of home. Come on up. Welcome. Welcome to you again. Thank you. By the way, best hair at the wine celebration, just saying.
Well, good morning, everyone. It's a real treat to be here, and it's also great to follow the 2 acts I just followed, because now you're all experts in immunology and cancer vaccination. So that makes my job a good bit easier. So what I wanted to talk about today is therapeutic cancer vaccines and what's going right and what's going wrong. And as you just heard from Dr. Finn, who, by the way, is one of the reasons I went to Pittsburgh, and I spent 15 years there. And nonetheless, when I got there, it was my first week, and I'm walking down the hall, and one of the older MDs comes up to me wearing a bow tie and says, well, welcome to Pittsburgh. What do you do? And I said, I make cancer vaccines. And he said, oh, you're one of the voodoo people. It's not voodoo any longer. So all of these things are tested in the clinic, and all of them work to one extent or another. All you have to do to show the immune system what it's looking for is treat it like an infection, as you heard. So you can show a little piece of the protein made by the tumor, some of the DNA or RNA from the tumor. You can show it the tumor cells, and that helps the immune system get pointed in the right direction. You can also, anytime you kill tumor, radiotherapy, chemotherapy, ablation, you can get those tumor proteins released, and you can get some cells activated from that alone. It's just not enough. So what helps are my favorite cells. These are dendritic cells. You can see that big, beautiful, huge dendritic cell reaching one of its processes out to tell the T cell what to do. And so that's the little T cell. The T cell is going to kill the tumor. The dendritic cell tells it what to look for and what to do when it gets there. These dendritic cells are all through your body, they're all through your skin, and they're always sampling the environment. And normally, if there's nothing going on, they once in a while, they go to the lymph node and they say, everything's okay. If you see any of this normal stuff, don't do anything. And if you get an infection, if you get a cut, if you get a virus, if you get a bacterium, they get activated and they go off to the lymph node and they say, this is what you're looking for and this is what to do when you see it. So they're wonderful cells, and they're great at starting those immune responses and shaping the immune responses. So we learned from Olya, we learned from a lot of these other experts, and we started to harness these cells against melanoma. So this is a series of 3 clinical trials that we did a long time ago, and we tried different numbers of dendritic cells. The immune system doesn't actually work in a dose-dependent way a lot of the time. So, you know, it just needs to see the target. It doesn't, unlike chemotherapy where you need more to be effective, and now it's too toxic. You can give a few cells, a lot of cells. That wasn't really a differentiator. But you give a prime, you give a boost, you maybe boost again, and that is a way to get the immune response going. So as you can see in these clinical trials we did in very small numbers of late-stage patients, we could mostly vaccinate everybody. 13 out of 16 in the first trial, 9 out of 10 in the second trial. Pretty much anyone who gets a good cancer vaccine, you test their blood before and after, they have more killer T cells. And that's not enough. But once in a while, you can see 2 disease stabilizations and a complete clinical response. And I'm going to show you in the next slide what that looked like. So it's buying time. It's buying non-toxic time. And once in a while, 5 to some trials, 10% of the patients, actually the tumor melts away and it stays away for a really long time. So we know what they can do. We just don't know how to keep that going and do it for all the patients who receive the vaccines. Some long-term disease stabilizations, 2 to 3 years. It's better than the alternative. Remaining no evidence of disease. You had disease, the surgeon got rid of most of it, but it was disseminated throughout your body. You know it's coming back. So you got the vaccine, and maybe it didn't come back for years. So buying time in a less toxic way has been one of the benefits. So here's one of the success stories. So this is a woman who had had surgery, radiation, interferon alpha, and immune therapy approved for melanoma. Again, we don't know how to use it really well. It doesn't work that well. So she has happily growing tumor, and what you see on the bottom is what the pathologist sees, and that's the tumor growing really well. After the vaccination, in her case, those lesions in the arm started to flatten out, and all those little dark spots you see, those are killer T cells that went to the tumor and then got into the tumor and killed it. And so we went back at the end of the trial to get more tumor, and we couldn't find any more. And so she got to go over 10 years without recurrence, and that blue bandage on her arm means she gave us a whole lot more blood to study at the end of that trial. So what we learned from that is that there's a component of inducing autoimmunity that might help. If you induce immunity to one protein in the tumor, if you can spread that to another tumor protein, then you do better, that diversification of the killer T cell response. You can get waves of cells coming in and continuing to kill the tumor. That can be a good sign. So we took some of those good signs and we did a lot of experiments. We went back to the lab, we applied for 10 grants, we got 1, and then we were able to find out what went right and what went wrong and design the next vaccine trial. So this was the successor trial to those. More patients this time, 35 patients. We did a randomization. We added interferon alpha to the vaccine to see if those 2 would work together well. And then instead of 1 tumor protein, we targeted 3. So we thought maybe if we need to get a snowball going, if we start with a bigger pack of snow first and then start that immune response, maybe we'll get a better immune response. So that's the trial we're analyzing now. And for the previous trials, we had 5 tubes of blood, and so we had ideas in our head from the literature of what we would ask. Did this matter? Did that matter? And we would ask pointed questions, and then we would answer those with the blood and the tumor cells given by the patient. And now technology has caught up. Now you can sequence everything. You can sequence it today, you can get the answer tomorrow. All of these wonderful technologies now, in addition to answering the questions we asked, we can answer the questions we don't know we even have. And we can just compare the blood before and after, tumor cells before and after, and let them tell us what we need to do next and what went right and what went wrong. So for melanoma, this is where we are, looking at all of these data and applying new technologies in my lab to answer those questions. So we did the same thing in liver cancer. It worked in melanoma, at least to an extent. We're learning a lot. We did it in liver cancer too. So we did different vaccines with peptides, with those beautiful dendritic cells, and we asked those same immune questions at the end. And what we saw, again, was we can pretty much vaccinate anybody against anything, even when it's your own cells and your own tissue. But here, we saw almost nothing clinically. We saw, once in a while, this tumor protein made by about half of liver tumors, alpha-fetoprotein. This is something that's in your blood when you're in fetal stage. It's turned off when you're born, but these liver tumor cells crank it up again about half the time. So that was our target. That was tumor-specific-ish, and so we went after that to generate the immune response. We can generate the immune response to that, but once in a while the tumor AFP in the blood, a measure of how much tumor, it would dip a little bit and then come back up. So what's wrong? Why isn't this working at least as well as it did in melanoma? And so we went back to the cells and we looked at the cells. And so there you see on the left the beautiful dendritic cell. They're not really those colors. And then on the top you see the experiment we did. The top blue is the nucleus, red is the cytoplasm of these cells. The bottom is electron microscopy. So on the left, control, beautiful dendritic cells. They're big, they have veils, they can point to the T cells and tell them what to do. If they've seen normal cord blood AFP, alpha-fetoprotein, they don't look so good. And the cells that saw tumor-derived AFP, it's not even the same cell anymore. And if you put it in a test tube with T cells, it doesn't activate T cells anymore. So now, what's the problem and how can we counteract the effect of this tumor? The AFP, it turns out, shuts off the metabolism of these cells, and they can't function anymore. And these are new technologies and new abilities to ask these questions. So we're plugging away in the lab. We're figuring out what to do about that suppression of AFP, the extent of it, how it operates, so that we can then counteract that. So in the lab, every year we find new discoveries, we make progress, we learn new things, we try new things. But when I was sitting in the back of the Society for Immunotherapy of Cancer meeting, and the founding CEO, Jeff Bluestone, got up in front and announced the existence of the Parker Institute for Cancer Immunotherapy, he said that this is going to be an institute that is laser-focused on cancer immunotherapy for solid tumors as a nonprofit research institute that would catalyze collaboration. So these are the focus areas: cell therapies. We know those killer T cells can get rid of the tumor. How can we engineer them better? How can we counteract all the negative effects of the tumor and make the cells resistant to that? Checkpoint inhibitors. This has revolutionized therapy for melanoma and for lung cancer. But they don't work yet for everybody, and sometimes people do recur who it looked like had a good response. So how can we figure out what works and what doesn't there? Tumor antigen discovery, you heard about the discovery of MUC1, alpha-fetoprotein. What else can we target that's different about the tumor that'll be even more successful? And then the tumor microenvironment. This is a very complex network of the tumor talking to the immune cells talking to each other, all in this, sometimes it seems like an impenetrable mass. So how can we then hack that to allow the T cells to get in? So these are the areas of focus. And then this is how the Institute operates. The network of academic investigators all get together twice a year and share unpublished data. And then the Institute partners with new biotechs and pharmaceutical companies and has flexible legal agreements. So instead of one university having to have 3 agreements with all these different people, each one has a single agreement with the Parker Institute. And then we can start pairing people together and put the combinations together that otherwise perhaps couldn't be done. And then at the hub, we have bioinformatics and data science that's phenomenal, and translational suite of all of these technology assays that we can then do with these trials. And we've got 3 different trials going on for diseases, and within each disease, there are 3 different trials going on, all at the same time, asking these combination questions about vaccination, about pairing, what order do you have to give these signals to the immune system to be most effective? So what's the solution? I think these people know. These are the next generation of young investigators that we're working together to support. And so far, the Parker Institute has spent $12 million to support this group. We have supported 5 or 6 a year of these young investigators. We put them in the network. We pair them with good mentorship and a great environment. And so I'll close by just thanking again the V Foundation for this symposium and the opportunity to collaborate again and support more of these new investigators who are going to learn from everything we've been developing over all these years and take that to the next level. Thank you very much.
Thank you, Lisa. And I love how you ended that in particular, because that's what we're so proud of at the V Foundation, is to bring up that next generation and continue the family feel of it, but the education, the communication, the knowledge, the research, that all ends up being tied together so this next young group can come and help us get to where we need to be is priceless. Thank you. Fourth, finally, the Isadora Lamp Professor and Chair of the Department of Radiation Oncology at the University of Michigan. We discussed this the other day, and I have to introduce you again now. Dr. Ted Lawrence, welcome to the stage. Thank you again. Thank you.
Yeah, there it is. It's great to be here this morning. And you're going to hear some, even though I'm going to come at this morning from an angle of radiation therapy, you're going to hear a lot of recurring themes from all the talks that will come back that I'll summarize at the end. So I'm going to cover a number of topics today in about 10 minutes. The first is in improvements in radiation therapy. I started this in my 1-minute interview that I had with USAGE Thursday evening on how radiation therapy is getting better and better with the addition of chemotherapy with or without surgery and with improved technology. Now I'm doing this, I haven't used the word immunotherapy yet in this part because the truth is the V Foundation hasn't spent much time thinking about radiation therapy, but this morning you're going to. Isn't that an amazing thing? And then I'm going to, of course, focus on combining radiation therapy with immunotherapy. And I'll give you a little bit of biology and then some very interesting clinical examples that are going to follow up on what you've heard before. Sometimes this works, sometimes it doesn't work. Why is that? But definitely a lot of excitement. And then I'm going to end with some really cool laboratory work from our group that's being translated into clinical trials that I hope amuses you and excites you. And all this in about 10 minutes. Can you get a better deal than that? I mean, what can you say? So radiation therapy alone or combined with chemotherapy can cure cancers. There's a rumor out there that I've heard from some people that when there's nothing else you can do, send the patient to radiation therapy. But we cure tens of thousands of patients a year with radiation alone or with radiation combined with chemotherapy. And I've listed all of the diseases here. And actually, I've organized my talk to spend about 20 minutes on each of these disease sites so you can really understand. Okay, I won't do that. I won't do that. But all of these are curable with radiation, or particularly radiation combined with chemotherapy when the tumor can't be removed surgically, so locally advanced cancers. And with surgery, combined with surgery with breast conservation, I think everyone in the audience must have heard that you can have lumpectomy plus radiation instead of a mastectomy for breast cancer. So thousands and thousands of women today have been able to preserve their breasts because of the use of radiation therapy. And the radiation therapy also plays an important role in chemotherapy in the treatment of pancreatic cancer. Since that's my research area, I just wanted to show you one example of how the combination of chemotherapy and radiation can do amazing things for patients, even with unresectable cancers. So what I'm showing you here, I don't know how many of you are used to looking at CT scans, but imagine the patient's head is in the wall here, their feet are out at you, this is their back, this is their front, that's right, that's left. Are you all reasonably oriented? And we've got a slice of somebody here. So this was the initial patient. And this is a pancreas cancer right here. And you can see these two little dots here, those white dots. I'll try to use the laser on it. It doesn't show very well. Right here, these are two key blood vessels. And this is the tumor right here. And this tumor is partially eating those blood vessels. When the surgeon looks at that, the surgeon goes, I can't do anything. I can't take out those blood vessels along with that tumor for the patient to live. Those are key blood vessels that are required for that patient to survive. And then after chemotherapy and radiation in one of our early clinical trials from many years ago, I hope you can see this has gotten quite a bit smaller. Here's the tumor, and now it's smaller, and those blood vessels are now much more visible. And now the surgeon is able to go in and remove that tumor. Oh, one more thing I need to point out to you. Can you notice here, this is the patient's abdomen. Do you see how that's all bent in? That patient's lost 30 to 40 pounds before they saw us in our clinic. And now can you see here, the patient's gained back that weight during the course of treatment, during treatment. So that's telling you that the treatment itself is not causing that much in the way of side effects. It's actually helping the patient quite a lot. So this is what happened because of the surgery. This is what it looked like beforehand. All of these are very, very, unfortunately, healthy-looking pancreas cancer cells. But now after surgery, after chemoradiation, and then the surgical removal, this is all scar. These black dots you're seeing here are dead cancer cells. So this combination, this is just one example. I could show you thousands of examples of how chemotherapy and radiation can kill cancers, either on its own or in combination with surgical approach. Now, the next thing I wanted to mention very, very briefly again is how improved technology in radiation therapy has had a tremendous impact on how we can cure tumors without hurting patients. So, the truth is, I'm old. I've gotten old. I don't know how it happened, just day by day, but somehow it happened. So this is what we used to do when I was a resident back 100 years ago. And what you're seeing there is a plain X-ray, and that crosshair is showing where the radiation is going to be aimed. And we just treated with big radiation fields, one treatment from the front, one treatment from the back. I promise you the tumor's in there, but if you're looking at that, so is a lot of normal tissue. So we couldn't cure many cancers because the normal tissue just couldn't tolerate this, and this caused a lot of side effects. Now, more recently, we have something called intensity modulated radiation therapy. I give 40-minute talks on this. I won't do this to you today, but the basic idea is the radiation can be much, much more focused on the tumor, and it's much better guided by all this modern imaging. If David Pinnick-Worms could give you a talk today, he and I would talk side by side about how the imaging of cancers has partnered with the treatment of cancers to produce much more focused and much better results. But what you can see here in these brightly colored lines is that's showing you the focused radiation on that lung cancer that, along with chemotherapy, is going to have a much better chance of curing that patient. And then this last panel, you're seeing stereotactic body radiation therapy. Again, I won't go into the details, but it's a way of using many, many radiation beams to focus very, very tightly on that tumor. And here with radiation alone, we cure tumors as well as surgery, non-invasively. The patient just lies on the treatment table, just quietly breathes, and that tumor is going to be cured. So the technical advances since I was a resident are truly spectacular. Now I'll actually turn to the topic of the day, which is, I hope you all are going to leave here saying, yay, radiation therapy. That was the goal of that part. But now I'm going to return to radiation and immunotherapy. And this is also an extremely exciting area. So in one slide, here is the biology of radiation therapy and immunotherapy. So radiation-induced immunomodulation. And this part is inside the cancer cell. So very simply, radiation therapy causes DNA damage. And the DNA is symbolized by these ladders, that's double-stranded DNA, and radiation's breaking it up. And that's happening inside the nucleus. This is the way radiation therapy kills tumor cells. But what also happens is the DNA leaks out of the nucleus and goes into the cytoplasm. And as the DNA leaves the nucleus, it mimics a viral infection. The cell thinks it's under a viral attack, because that's what viruses can do. Their DNA double strands inside the cytoplasm. And that can activate an immune response. One of the areas of research that we're very excited about is adding DNA damage repair inhibitors. You can imagine if you inhibit the repair of that DNA damage, combining that with radiation can sensitize to immunotherapy by producing even more of those. And it can produce something called an abscopal effect. That's a fancy term we use for saying that what you're doing to that tumor with radiation can activate T cells, and as you heard about this morning, which can circulate through the body and potentially attack cancer at a distance. And we think we see that in certain circumstances that we'll talk about a little bit more toward the end. Now this, the way I presented it, it's all radiation should alone activate an immune response and cure all cancers, but unfortunately that's not the case. And what I'm showing you here is this seesaw, that there are some positive effects in stimulating the immune response with radiation, but there are some negative effects as well. So the positive effects, and you've heard a little bit about this already, the first I have here is antigen presentation. That means when you radiate a cell, that cell now shows new targets for the immune system to recognize. You also get chemoattraction. What that means is that actually in the previous slide, I should have mentioned that. See if I can go back. There we go. You get something called type 1 interferon. So the type 1 interferons draw cells, the immune system, to that tumor cell. And then that attracts those cells and can stimulate the response, and those cells can then go out and circulate. In addition, it makes those cells angry, so you get increased CD8 T cell activity. So all these things, you're bringing in the immune cells, you're making them angry, you're permitting them to circulate. These are all good things. But you also get an expression of something called PD-L1, which is a normal antigen, a normal protein on the cell surface that the normal cells use to tell the immune system that they're a friend. Cancers are so tricky. They use this and say, I'm not your enemy, I'm your friend. They put this out toward the immune system. And so this gets stimulated by radiation as well. So you get positive effects and negative effects. The cancer can use some of this to hide. And what that now is, as Bill mentioned earlier, there are now agents, there are immune checkpoint blockades that can cover up that PD-L1 expression and make immunotherapy combined with radiation therapy work even better. This is an area that's under research, but there are positive effects and negative effects that both can happen, and we have to learn how to work with these. So this is the biology. So let me show you some clinical results that really highlight some of the things that you've heard already. There are negative results and there are positive results. So the first trial that I'll talk to you about is a 62-patient randomized trial for patients with metastatic head and neck cancer, and they were treated with nivolumab, which is one of these immune checkpoint blockade inhibitors, one of these ones that covers up that PD-L1 and makes the tumor cell more visible to the immune system. And these patients were randomly allotted to have stereotactic body radiation therapy, that very focused targeted radiation, to one cancer with the idea, would it activate the immune system by attacking that one tumor, and then would it affect all the tumors in that patient's body? Unfortunately, that result was no. There was no evidence of increased response in lesions not receiving the stereotactic body radiation. But then we have another example of a 76-patient trial with metastatic lung cancer, patients with metastatic lung cancer treated with pembrolizumab, and not on one of these PD-L1 inhibitors, plus or minus stereotactic body radiation to one lesion. And here the overall response rate was doubled with SBRT from 18% to 36%. And the overall survival was doubled from 7.6 months to 15.9 months. So these two very different results. Why is that? Well, one is they're different diseases. Not all diseases are the same. As been mentioned, lung cancer might be much more sensitive to immunotherapy. Head and neck cancer, not so much. They use different radiation doses. They use different immune checkpoint blockade inhibitors. And both of them are relatively small studies. Now, I want to show one more data slide here, just, not really data, I have a couple more seconds. Oh good, Bill's shrugging his shoulders. I got another half an hour. Now, this is a very, very cool study that's about to be launched at Michigan that I'm very excited about. And it shows you yet another aspect of immunotherapy. The liver is an immunosuppressed environment. If every time you ate a new food, your liver reacted to it, you'd be constantly, your immune system would constantly be going crazy. So the liver is a basically immunosuppressed environment. And what our studies have shown is that patients with liver metastases do worse. You have two relatively similar patients, one who has liver metastases and one who has lung metastases. The patient with liver metastases does not respond to immunotherapy as well as the patient with lung metastases. And what we found in this paper that we published in Nature Medicine earlier this year is that the macrophages within the tumor, within the liver, kill the good guy CD8 positive T cells. They scoop them up and kill them. And so the concept here would be, if we can kill those tumors in the liver with stereotactic body radiation, we'll get rid of those metastases that are getting rid of the good guys, the CD8 positive T cells that can kill the tumor. And this will then permit those cells to go and do their job. And we have partial funding for that, and we're really hoping to launch that trial later this year. And this is a unique approach to using immunotherapy plus radiation therapy for cancer treatment. So you see that's glow, that's not radiation, is it? I hope at this point in my talk, you're thinking that you're not quite as nervous as this young girl. You know that radiation therapy can be very helpful. With chemotherapy, it can cure some cancers. Particularly with surgery, it can cure cancers. We have to be careful with it. You've got to be careful. But maybe not quite as nervous as this little girl. And that radiation therapy can be that fairy godmother that goes around and cures more cancers around the world. So in summary, radiation therapy plus or minus surgery and systemic therapy cures many, many people with acceptable side effects. And that radiation therapy combined with immunotherapy is promising, but really it's in its infancy, just as what you've heard this morning. And the successful combination is going to require better understanding of which diseases and the specifics of treatment. And this is just the kind of thing that the V Foundation is supporting in the research that you support for our fellows at the earliest stages and also our translational researchers. So thank everybody here for all you do for the V Foundation, for supporting this kind of research, and we're very excited about the future because of what we've seen today. So thanks for your attention.
Thank you. Thank you. How about the positivity in this room? Despite a serious topic, I mean, there's so much reason to be positive and optimistic, I think, about the future, for sure. Yes? I mean, especially when I have this guy next to me. Who's more positive than Dr. Bill Nelson?
Hey, many of you guys like sports like that. I've always dreamed of hosting a program with an ESPN.
No, you have not.
Absolutely.
You're so
I thought we'd be
full of
doing top 10 plays or something.
Oh, how about the not top 10? I'm very good at that. Yeah.
But what we're going to do is entertain some questions,
Yeah.
and we have all of our scientific advisory council members here. And so, because of the way we're spread out, socially distanced today, is Sage is going to, they're pumping the questions right at her, and then she's going to throw them over, and we're going to figure out who to
He'll
to
translate,
pick on,
right.
and then wave your hand, and they'll come to the microphone with you at the distance that you're at.
Yes,
Does that make sense?
yeah,
No, it
so
doesn't.
you don't need me, frankly. But that's the thing, is yes, the virtual audience, we are listening to you and feeding the questions in as well as you here. And for those of you here in person, if you look to your right, Marketplace, and for those of you watching, please prepare the vintner. Yes, you see that the wine continues. We must be in Napa or something. So that is happening right after this as planned. I want to make sure that you know that just because this part changed, that part does not. So more to come on that in a few minutes. Oh, I see questions being typed, which means this is live and in person, right? Oh gosh, that seems like that's a big broad one, right? So I'm going to be glad to read it and then step off the stage and good luck.
That's called a throw in your business.
That's called good luck. Bye. Peace out. But this is. Excuse me? Yes, audience questions are coming, but I'm starting with this one first. And I think it's fascinating because it sounds great. Fingers crossed, I hope what your answer is, but it's going to take some work, right? Can we give an overall cancer vaccine someday that will prevent all cancers? I mean, that's the goal, right? How realistic is it that we get to that?
So that kind of question, obviously, somebody was listening to Olia Finn's talk clearly, carefully, and I think we should throw that one right in her lap. You're trying to prevent some cancers. Will there be a single vaccine that prevents them all?
Working on it.
Okay.
Yeah.
The positivity.
I have an NCI grant that is a 7-year grant, and it's called an NCI Outstanding Investigator Award. And I got it 5 years ago, so I have 2 more years. And it had 4 goals. And my fourth one, I called my audacious goal, to create a universal cancer vaccine. So Mach 1 is close to that because it covers over 80% of all human cancers. So that could be enough. And there are a few others like tyrosinase, H3, and right. So there are a few other antigens that are in that category that could cover a lot of, at least a lot of most solid tumors versus lymphomas. So we can divide them into categories. So I think we have multiple candidates. The one that I'm working on is a brand new idea where early in your life, when you're exposed to childhood diseases, febrile viral infections, your immune system gets trained to recognize a sick cell and to get rid of it. And sometimes faster than knowing what made it sick. So the important thing is to get rid of it and not take the time to figure out why that cell is not well. So you have this memory, deep memory response that was generated when the immune system was young, had a lot of naive cells that can be educated, and it's in your deep memory. So when there are many other viruses that you're exposed to that you don't even know that you're exposed to because the very first infected cell was recognized and gotten rid of, a lot of the molecules that the immune system recognizes on sick cells are also present on cancer cells because it's a sick cell for a different reason. It's not infected, but it's mutated, etc. So I am compiling a list of those molecules that are shared that I call disease-associated antigens / tumor-associated antigens. And in animal models, in mouse models, we show, for example, that repeated infections with the flu virus in mice can generate an immune memory where 9 months after the last infection, you can give a mouse a tumor and it will reject it, whereas flu-naive mice who never saw the flu will die of that cancer. So this is one idea that I have been pursuing for several years now, and there's epidemiological evidence that people who have had multiple infections early in life have reduced risk for cancer. We have done some of that work ourselves, but there's a lot of work out there that epidemiologists have found out that there's a tremendous correlation with risk reduction and previous exposure to febrile viral diseases. So if we can focus on those shared antigens, perhaps we can tap into everybody's memory generated in childhood to protect you from cancer later in life.
Wow.
Did everybody get that?
The positivity, once again.
Continues, that's
Yes,
right.
absolutely. Thank you, Dr. Finn. And you might be called on next, so again, so don't get too comfortable. We might be coming back to you. Can all types of cancers be treated by immunotherapy?
So yeah, there's been these incredible successes against lung cancer, against melanoma, but people still get breast cancer, they still get prostate cancer, they still get these other diseases. What's the state of immunotherapy? That's sort of that question. I'm going to pick on two people for this one. First, on Nancy Davidson. Breast cancer did not melt away to our early immunotherapy, but it does seem to be some progress. What about immunotherapy for breast cancer? Nancy's one of the great breast cancer experts.
Thanks so much. You are right, Bill. We wish we had a better response to immunotherapy in breast cancer. But you know, one of the things we realized is that there's a lot of different kinds of breast cancers. And so we need to subset them. And it looks like, at least right now in the earliest studies, that the kind of breast cancer that's called the triple negative breast cancer, that's the breast cancer that doesn't have estrogen receptor, it doesn't have progesterone receptor, so it's not very sensitive to our hormone therapies. It's a kind of breast cancer that doesn't have the HER2 protein, so we can't use our anti-HER2 therapies. But it turns out these triple negative breast cancers might be more susceptible to some of these drugs or agents we've been talking about, the checkpoint inhibitors. And in fact, in the last couple of years in the United States, we've learned that these triple negative breast cancers, in their most advanced form, can be treated with immunotherapy, with checkpoint inhibitors, perhaps with chemotherapy. And just in the last month, actually, the FDA has approved the use of one of these checkpoint inhibitors in certain situations in early breast cancer, newly diagnosed breast cancer, where we want to use chemotherapy now with this immunotherapy as a preoperative form of therapy to shrink the tumor, to make it easier for the surgeon and the patient to go through surgery, and then continue it afterwards to try to mop up any residual cells. So I think this is a real clear example of having to take a really big cancer, break it down into the various subtypes of cancer, and then think in a very precise manner about where you can use immunotherapies. And, you know, trastuzumab, anti-HER2 therapy, that's a different form of immunotherapy. There are plenty of them out there. So this is such an exciting time because we've realized that we have to do this reduction of big diseases into smaller diseases where we can find these more precise approaches. More to do, but certainly the first inkling of success in breast cancer with immunotherapy.
So could you pass the microphone over to Mike Caston, because he was the next one I was going to pick on. Children with cancer have led the way in producing some of the earliest cancer cures. Some of the drugs we conjure up for cancer seem to work their way into clinical trials for children kind of late on in the game. But immunotherapy in children, CAR T cells, what's the state of immunotherapy in children's cancer?
Well, as Nancy discussed at breast cancer, the checkpoint inhibitors, the inhibitors that are activating the immune system that have been so successful in non-small cell lung cancer and in melanomas, have not had the same success in childhood cancers for reasons that are not totally well understood. But as you just pointed out, Bill, there has been an extraordinary success in using immunotherapy to treat childhood cancers, and that's with CAR T cells. So the big excitement a few years ago was using CAR T cells that target the B cells in the immune system that cause acute lymphocytic leukemia and lymphomas. They are exquisitely sensitive to CAR T cells that target one of those specific tumor antigens on those cells. So these children who had relapsed every type of chemotherapy and radiation therapy that had been successfully used in the past, and these children were within weeks of death from their leukemia, CAR T cells turned out to be curative in them. And so this is being expanded to many types of leukemias and lymphomas in children and also in adults. There's still a tremendous amount of work going on to understand why the immune checkpoint inhibitors don't work as well on childhood cancers and try to turn that around so that we can manipulate other aspects of the immune environment and the antigens that the tumor cells are presenting to make them more sensitive to these immune checkpoint inhibitors.
And of course, Sage, you know, the V Foundation has had a long-standing interest in cancers in children.
Absolutely.
Investing in V scholars and translation research, and Michael heads up the review of that event.
And what Dick Bartel does at his annual event, and he's raised so many millions of dollars specifically focusing on pediatric cancer. So thank you. And real quick, I want to ask the technical crew, I don't know if that's a live camera, can the people at home or watching virtually see? Do I need to have the doctor stand and face that way? Yes. So
Face
next
the
time we
camera
call
back
on you, please stand and turn to the camera. I know you'd rather look at us all day, but you need to look at the cameras to make sure everybody virtually can enjoy all of it and get as much information as possible. So thank you, thank you. We continue. Can we donate our cells to create our own immunotherapy vaccines if we've had cancer?
I think this one I'm going to pick on Richard Fisher, actually. You know, we heard a little bit about donating cells. If you have cancer, they'll make them into CAR T cells. The adult footprint of that is oversampling at the moment. Cancers in the B cell lineage, myeloma, and whatnot. Something you know a lot about. And we treat these people. Sometimes it's easy to yank out enough cells. Sometimes it's not. There may be other tricks, bispecific antibodies. I've thrown enough at you that you can come up with something to enlighten all of us.
Just a microphone. Yeah, you can take it to him. That's fine.
Yeah. Oh. There we go.
Well, thanks for that question, Bill, I think.
It's actually 20 questions or something.
I know.
Yes, I'll be done in about an hour. No, seriously. So the excitement in the hematologic malignancies and in lymphomas, which you heard a little bit about from Mike, in children is also in adults. So what you see is that patients are cured in many of the adult lymphomas by chemotherapy. And this actually gives us a problem we need to think about, which is when you bring in other new types of therapies, how do you back off what already works? If you're curing 80% of the patients in Hodgkin's disease, 90%, how do you bring a new therapy in there, and how do you get it up front? So what you do is you look at the 10% who aren't doing well, and you start there, okay? And the immunologic approaches there are very interesting. One, checkpoint inhibitors, and they work in Hodgkin's disease, but darn it, they don't work at all in large cell lymphoma, and we don't understand why. So again, as Nancy said, very specific treatments. But what came out in the CAR T is very interesting. So CAR T is an immune cell that recognizes an antigen on B cells. To date, the majority of them are CD19. That's just something sticking out as you could be a spike protein, but it's not that. It's an antigen on these cells. And what happens is that the immune therapy, these CAR T cells, kill any cell that expresses the CD19. And it turns out you can live without CD19 cells, and you can deplete them very strongly. So we're moving forward, and in those refractory cases, we're getting very good results. And in fact, we're getting results where 40 to 50% of all of those relapsed patients are actually cured. Appearance is maybe, let's say, 5 years free of survival. That's great, right? So that suggests they're cured. Now you begin to say, what else can we move that up into? How do we take away some of the toxicity of chemotherapy? And one of the interesting things that's going to happen is, you probably all know that at end stage, not necessarily end stage, but close to it, bone marrow transplant is done. And that's a very intensive, go in the hospital, get high doses of chemotherapy. And what you're going to see now is the results are so good in CAR T therapy that in large cell lymphoma, aggressive lymphoma, we're going to run a randomized trial comparing bone marrow transplant versus the CAR T. Now, I got to end with some reality testing here because someone's going to jump on me. CAR T is not without toxicity. Okay, that's a problem. And what happens is when you whip up the immune response, you get a really sick patient in most cases. Some look a little less, some look a little more, but it's a real tough toxicity. And there's what's called a cytokine storm. All of these inflammatory molecules come out, and they can make you really sick. So it's not a free lunch yet, but it's something we're working on. And I'm not sure, did I answer that question?
I think you answered at least 10 of the 20. I'm just kidding. No, you didn't.
Thank you. I would like to mix in a couple of questions from the people who are here. Julie, do you have a question? Thank you.
That was perfect
so Julie,
timing.
I have to have you stand up. Stand up,
okay,
look at the camera.
Yeah, so that was perfect timing because my question had to do with exactly what your, Bill Nelson and I had a conversation about this earlier this year, which is when you have somebody with leukemia in aggressive form and they have to find the bone marrow match, right? It can be a needle in a haystack sometimes. And I know that there is a young investigator who was funded by the B Foundation who's looking to create a universal bone marrow transplant. And I'm curious, does that then, what do you think about that? Does that then make that the best possible case solution where you're going right to that? Or how do you see it? And is that a reality?
Sorry. I think we need two runners.
Julie, that's a complicated question, and I wish it were really a 30-second answer to that. You know that there are sometimes bone marrow transplant where the rescue cells, bone marrow transplant is just a lot of chemotherapy and radiation that you couldn't survive unless we rescue you. It's really a simple concept. So you take out your own cells, put them in the freezer, give a lethal dose, but then you rescue the patients. If the cells come from somebody else, you have to match them because, in fact, the immune response will reject mismatched cells and you'll get sick as anything, okay? So what we're having is national donor matches, and they're very important. And by the way, one of the issues is that the minorities and the underrepresented minorities, people of color, are underrepresented. So they have trouble finding a match. So if someone could find a universal match, that would be great, and it would be well done. I think we're not there yet. We're looking at that, and there's studies going on, but it's a tough question. Now, what I'm saying is, as opposed to doing that, in some of those circumstances where you do transplant, you might be able to use a CAR T cell, which is the patient's own cell transfected with the right material to make it go against the tumor, and we might not need to do such toxic therapy. And that's the question. And those will balance out over the next couple of years.
Thank you. We'll go back to the questions coming in from the virtual viewers. Yeah, fair question. What are the possible side effects of immunotherapy, Dr. Nelson?
Yeah, we're still looking for the miracle cure with no side effects and cure everyone, but there are side effects, and we heard about some of them.
Yeah.
The cytokine storm, very similar to what happens to some people with COVID-19. And one way to look at it is any organ you can name and put the suffix itis on it is in play. And I was going to pick on Shelley Earp because as cancer center directors, we've had to build systems to accomplish surveillance of people on immunotherapy. So if they have one of these itis things, we can work with them, get them the right health care and stuff. So you've thought a lot about this. You have to.
But that's about good clinical practice. We do clinical research. You have the first CAR T, you have the first immunotherapy, you do these trials. We're all together on this. There's a little bit of competition in cancer research, but mostly there's cooperation with learning about patients. And so over the last 7 or 8 years, we've seen a couple of waves of what are called these immune checkpoints. The first one was actually more toxic. It was anti-CTLA-4. The newer generation is a little bit kinder and gentler. But as we have looked at it, virtually, as Bill said, any organ can have an itis, because what you're doing with CAR T is you're revving up your immune system. Now, earlier in our lives, we educated our T cells not to attack ourselves. And when we give them this anti-PD-1, we're kind of diseducating them because we want them to go after the tumor cell, which is really part of ourselves. But sometimes they start to go after normal tissue. The one that happens the most is the thyroid. That's okay. Trade-off for curing your lung cancer, taking thyroid for the rest of your life, that's a pretty good deal. But you can get kidney disease. You can usually handle that with some immunosuppression, which turns out not to affect the anti-PD-1 efficacy against your tumor. Lung fibrosis is a little worse, and we worry about that. So you have this series of things. But we're also very much looking at these. Now the second wave, okay, we know these things. Cytokine release syndrome we just talked about. Well, one of the things that we're doing at UNC is we're putting actually 2 genes into the CAR T cell. The one recognizes your tumor. The other one is called inducible caspase 9. And what that does is have a gene that's poised to actually kill the CAR T cell. But it won't work, and you don't want it to work. You don't want to kill the CAR T cell. But if we give the patient a drug that turns that second gene on, we can get rid of the CAR T cells within 24 hours. And we've had some cases where cytokine release, we're learning how to deal with that. but there's some brain neurotoxicity that we don't quite understand. And we've been able to give this drug and actually get rid of the CAR T cells so that you get rid of the toxicity. Well, gee, then what happens after that? Because you actually want the patient to survive and you want them to get rid of their cancer. Well, we've made billions of these CAR T cells. They're in the freezer, so we can re-challenge the patient. This is the iterative process of translational clinical cancer research of the type that the V Foundation is actually going after. So thank you for that support.
Thank you. I think one of the concerns that has been going around since the pandemic began is wait a minute, what about cancer patients and people who continue to need treatments, but you know, the effect on that, even just the logistical part of getting into these treatment centers and hospitals. How much have you, and anyone else who wants to chime in, dealt with that and the concerns from your patients, and how to make sure they still get the best care, because cancer doesn't wait?
So we've asked, you know, our whole society to do a lot of things, right? We wear masks, we do social distancing. None of this stops people from getting cancer.
Right.
Right, so we need to get them the help they need. I was going to pick on Bob Bass for this. The MD Anderson, I know it's a small community hospital in Texas, but people travel great distances to go to the MD Anderson. And people, I mean, you hear those three words, you have cancer at a time when you can't go travel, you can't get a mammogram, all this kind of stuff. What's been the disruption you've seen, and what kinds of adaptations are our nation's cancer centers making?
Huge problem, and one that we'll be dealing with over these next year or two, or perhaps beyond. Certainly part of it is diagnosing cancer up front and getting the regular colonoscopies that everybody needs if they're going to detect cancer early when it's curable. Also, certainly getting the kind of consultation that you need is a challenge. And if you do get COVID, getting through that if you've got cancer. And certainly one of the challenges that MD Anderson took on very early, given the fact that we care for 100,000 cancer patients at one stage or another of their illness, was how we could get cancer patients through that as safely as possible. It turns out that we sent all of our staff home who didn't deal with patients. So we've been working remotely for the last year and a half. We've also were compulsive about anybody coming in getting a COVID test. We also geared up with 3 or 4 different stages of observation for people with COVID or suspected of having COVID. So we had different wards set aside for that. And we ramped up our intensive care. You know, with those kinds of precautions, we had only 3% deaths of cancer patients from COVID at MD Anderson. That compares with about 21% internationally in about 20 different studies showing that, as you heard earlier, patients with cancer are more susceptible to COVID. Those kinds of best practices really can be instituted widespread, provided the health system remains in balance. And as you know, over the last year, with the 2 or 3 or now 4 different waves, we stress our health system to breaking point in a lot of different communities. I think it just underlines, and I know everybody here is vaccinated, but I think our challenge over these next months is to see more and more people who could be vaccinated, who are willing to be vaccinated, but are just holding out a bit, get across that finish line, and that we end up with enough immunity that even with these more rapidly proliferating viruses, we do better.
So I wanted to double down on that a little bit with you, Fumi. You've spent your entire career trying to get ahead of the game. Use genetic testing, genetic screening, find out who's vulnerable, and intervene so they don't get these terrible cancers, breast cancer in particular. How are we doing that? That's a high-end kind of thing when people are, you know, masking, hiding under the desk like me, slathering your body with hand sanitizer. How do we do that? And they're vulnerable people who are even more vulnerable now.
Yeah, so one of the things we learned during this pandemic is that we can actually do better by going to our patients' homes And so to follow on what Dr. Bass said, we closed our hospitals because most of the hospitals were filled with very sick COVID patients. And we saved a lot of lives by doing clinical research and saving them with good clinical care. But then we quickly realized that if we don't get high-risk patients to come in and get their mammogram, we've prevented them from getting COVID, but we haven't addressed the risk of cancer. So one of the things that we had to do was to really get genetic counselors who do genetic testing to embrace telehealth, right? All of us who were over the age of 60 had to go home because we were at high risk of dying from COVID. Imagine me staying at home and doing nothing. That's not possible. So I became a Zoom doctor, right? We have virtual visits, and we immediately transformed our ability to get genomic tests to the patients. Doctor's choice, right? We send the phlebotomies to go to our patients because in all honesty, cancer is worse than COVID, and we knew that. And so for what we are talking about in terms of getting ahead of the game, so many women, so many men have missed the opportunity to get their colonoscopy, to get their mammogram, to get all of their screening tests. Men have not gone to get their PSA. So how do we get the whole engine moving again? That's where we're really gung-ho about telehealth. If you can't come to us, we want to come to you. And that's really where we're hoping that after this pandemic, we will have a solidarity where it doesn't matter where you live anymore, because whether you're in rural America, you're in urban America, you don't have access to a doctor, we can send a drone to you, right, to deliver your drug. That's sort of the wave of the future. We're getting more and more drugs that can be taken orally. And we learned, for example, in breast cancer, that we thought everybody had to come to the hospital. And then we quickly pivoted, right? HER2 targeted therapy. I had a woman who had been on treatment for more than 16 years, keeping her metastatic breast cancer in check and coming to get an infusion. And it turns out we can actually do an injection at home that was going to be faster than having her come in. So lots of innovation had to come in. And I said to Olia, we're going to really find that vaccine, because if we identify high-risk patients, right, which we're planning to do now, is to prioritize those patients as the first to come in to get screening, because we're backed up, right? That's why I said know your risk. And then those who are not so much at high risk, then we start getting them back in. So we're moving beyond using age to prioritize who gets screened to using different levels of risk. And we're going forward, and this is really my favorite topic. So, you know, Milwaukee won the NBA Finals this year. And guess who was the tallest, most prominent guy there? A Nigerian who was 7 feet 5 inches, right? How did he get to be 7 feet 5 inches? His parents are not that tall. So we're really in the age of using the whole genome, looking at how all these complex traits are inherited. So he won the lottery by winning all the genes that code for height, right? And then he trained to be a really great athlete. And now Milwaukee won the NBA Finals. So that's also the question. Can we stratify the entire population into the high-risk category and then the low-risk category? And if we can do that for every cancer, you can imagine that we would get to a moment where we can get the high-risk patient their vaccine and they go on their way. And then the low-risk patient, they can drink all the booze they want, right? That's where we're going.
And I think it's all about the booze, isn't it? I will say it's beautiful, and maybe a small silver lining in all of this is the ability to think outside the box and say, okay, there are other ways to do this. One thing that I want to make sure we don't lose, though, is this, right? And sometimes, especially when you're getting a terrible diagnosis or you're dealing with the treatments, and the bedside manner that you have, I mean, that's important when people are afraid. So what's that line where someone might say, I know I have some risks, but I need to see you in person and look you in the eye?
I'm really glad that you brought that on, because, you know, while we embrace technology to make things better for all of us, we also know that medicine is about the humanity, about the touch, about the doctor who cares, right? And so, as we address how technology can help us advance cancer research, we're also spending a lot of time on the humanistic aspect of medicine, teaching art therapy, music therapy. And as you know, during this pandemic, a lot of artists were also at risk, and we had to mobilize some of the resources to actually help patients feel better even while at home. So I think that we're all in this together. What I hope will happen after the pandemic is over, and it will be over very soon, because we're going to get everybody vaccinated, right? And when we all come out of this, I hope we would all have learned a lot about our own humanity. We hear about black and brown people dying unnecessarily because they didn't have access. And I think that's why, as much as we talk about all the research that's going on in academic medical centers like MD Anderson, those of us on the south side of Chicago, those of us in Chicago, New York, all the big cities are also thinking about health equity and how we can mobilize our resources and get to people who are really hard to get to. So I'm very optimistic that this pandemic has taught us a lot, and globally we're talking about being in solidarity, and I think cancer research will benefit from all of us coming together to do better in the future. So I believe in Build Back Better, but for doctors and nurses and physical therapists, all of us who have been in the front line, the paramedics, the firefighters who fought during this pandemic, I think we learned something about how we can now use our energies to do better in healthcare.
And I thank you. And I'm so thankful for all the doctors here who seem to get that human side, right? Where thank goodness for the wonderful doctors at Johns Hopkins where my dad is being treated, right? And it's tough sometimes when, I mean, they're a pair, married 50 years in October. Thank you very much. Hi, Mom and Dad. And right? Amazing. And so, but they're partners, and they've been dealing with this treatment together, even though he has the cancer. So when they have to walk into those hospitals alone, without that person, it is on you, the doctors, to remember that human side, and obviously you do. So thank you for remembering that stuff too, while you're doing all the fancy science.
Oh yeah,
you know,
absolutely.
I mean, it's so important and it's emotional. So thank you.
So we were talking about this the other day. You know, cancer research, cancer care, they're a team sport.
Yeah.
And the thing that's exciting this year is we are winning. We're starting to win. And we were talking about that yesterday. It's starting to work. I think we'd be running up the score if you were 7 feet tall, but we're definitely winning. And being on our team is helpful.
Giannis and the Bucks did not run up the score. Yeah. They just dominated. Okay, we have a few minutes left. Anyone in here with a question before I go back to the virtual questions? Any hands raised? No? Okay, trust me, I have more. Specifically to Dr. Finn. How do we...
I think we can steer this one to someone else, actually.
Okay, okay.
So the question was, how do we get people into vaccine clinical trials, into clinical trials? And I had a chance to write a little piece for another thing from the chemotherapy era, where we tried to approve drugs that just blatantly kill cancer cells, and the question is usually, how much can we give people? Can we give them enough to kill cancer without hurting them too much? And from that era, it would take 12,000 people on average to participate in a clinical trial to get one drug approved. Now, the nice thing about the science that the V Foundation is invested in and the scientists, is this is getting much, much more efficient. And I'm zeroing in on Susan Hilsenbeck. We're getting to a stage where we're doing clinical trials. She's the scorekeeper. You have to have a scorekeeper, and we need to know if we're winning, if we're losing, if it's a tie game. And her discipline uses mathematics and other things to figure out, is it working? And we're getting better and better and more efficient at that. How is that working?
Is this on? This is a first. I've come to this symposium, I think this is my 9th time.
Wow.
Never gotten a question for the statistician.
And you look so comfortable sitting there. Yeah.
I love it.
So I'm thrilled to get asked a question. This is fantastic. So what Bill just said is that when we have kind of blunt force treatments that only really benefit a fraction of people, and we don't know which ones are going to benefit or not benefit, it takes a lot of people total to see that small difference in the fraction of people that benefited in the treatment group versus the standard treatment group. So there are breast cancer trials that have had thousands and thousands of patients in them to detect a 3% or 5% difference in the new therapy versus the old therapy. With targeted therapies and the kinds of treatments that we're talking about that affect specific subtypes of cancer, we expect almost everyone with that subtype of cancer to benefit. So we expect to see an enormous difference between the old therapy and the new beneficial therapy that specifically hits that target. That only takes a small number of people to see an enormous difference. So what I think about when I'm helping people design trials is, what do we expect to see? And then what's the minimum number of people that we need to involve in the trial in order to have a high probability of seeing that difference? And in very targeted therapies, or therapies like the vaccine trials that we're talking about, where we expect there to be, if we hit the target right, we expect to be a big effect, we don't need to study thousands of people. We only need to study maybe hundreds of people or tens of people in order to see that difference. And we're designing trials that are really focused on that kind of detection of differences. And even though it's a small number of people, they're very convincing because we see this enormous effect. Does that address the question?
Yeah.
I think we need to do this every year with you. Yeah. Be ready. Thank you. Last question. Why don't you begin?
So the question that was proffered was, which of the immune checkpoint inhibitor molecules are the most promising? Somebody who's asking this question knows that there are a lot of them working their ways through clinical trials. I thought I'd twist this around to a question for you, David Punicle-Worm. Most of these are antibody drugs, which means we can figure out where they went. It's his business. Are we close to an imaging solution to say, in this particular cancer, we could use this particular immune checkpoint inhibitor? Do you see us getting there? And if we're not there yet, what's going to get us there? By the way, you heard earlier, imaging and radiation oncology, they're tied at the waist here. They're partners. Is this going to be the same thing with imaging and immunotherapy, do you think?
And a look into the near future on that. So this involves the role of molecular imaging. And I want to make a distinction that there's sort of the standard anatomic imaging that many people are familiar with, and then there's molecular imaging. Standard anatomic imaging is the MRI, magnetic resonance imaging, cross-sectional imaging like the CT, CAT scans, computed tomography, et cetera. So MR, CT. And we saw an example of that in an earlier lecture from Ted. The anatomic imaging is very important for identifying the anatomy. Where's the tumor? Does it locally invade? and importantly, the collaborations between the imagers and the radiation oncologists on designing the new approaches that Dr. Lawrence mentioned about how you shape radiation beams. But it's about anatomy. The newer type of imaging that's coming along and is now into the mainstream is looking at molecular details of that tumor. What are the receptors that might be on the surface, or what is the metabolism? And that can complement some of the genetic information that is now very commonly done. Biopsies are done to sequence and find out about the genes. And despite the excitement about looking at mutations in genes of tumors, not all the information that's important is necessarily contained in the gene and the sequence. A lot is, but a lot of that can be complemented then by having specific molecular signatures of what's on the tumor. And what are the technologies that do that? You've heard of them before, but they're expanding rapidly now. Positron emission tomography, PET scans. There's been one molecule that's had a major impact on cancer care, and that's fluorodeoxyglucose, which is a sugar that's radiolabeled with a small amount of radioactivity, fluorine-18, and that's injected into the body at safe levels, and someone will have a PET scan, and that'll often be used for staging. It can look at that specific metabolism of the tumor, and you can see where is the tumor and are there metastases. Because it's impossible to do surgery and explore everywhere. The genes don't tell you where the cancer might be, but you might want to know, is there a local breast cancer or a local colon cancer or a local pancreatic cancer, but then are there metastases somewhere else? Well, it ends up that the imaging with PET can help stage the tumor. And it's very different treatments if a patient has local tumor versus metastatic tumor. Very different pathways of how all the therapies will come together. Well, along that line, there's new molecules that are coming along to look at the signatures with these type of new PET imaging agents that are just on the threshold of coming out to help us not only stage the tumor, but also complement the gene sequencing, complement some of the histology, and to know then how to direct therapies to that tumor. And that's some of the new peptides and antibodies that are coming along. And there is a new field that's emerging. Again, it's been under development for 20 years. We talked about it briefly before, but that's actually combining diagnosis and therapy, and it's a field called theranostics. Thera meaning for therapy, the second part of it, diagnostics, so theranostics. And in radionuclide theranostics, you can actually have a pair of molecules that are often an antibody or a peptide, where then you can put a small amount of safe radiation on that with something like fluorine-18, and you can then again diagnose a molecular signature of that cancer and know that that cancer has that particular surface molecule. And maybe like MUC1, but many others. And then you can put on a therapeutic beta emitter or alpha emitting radioisotope onto that same molecule. And now you know that tumor has that target, and now we can follow it up with a beta emission. And there's an explosion of that activity that's coming out now, and just recently FDA approval of a pair of molecules now that goes after PSMA, a cell surface marker in prostate cancer, that for certain subsets of patients has had dramatic effect on curing those patients, or at least managing their disease that previously wouldn't have had other alternatives. So yes, there's an explosion of activity of some of these new combination therapies to look at molecular signatures through molecular imaging. And again, combinations I think are the key. These might well be combined with radiation, combined with immunotherapy, and much research on translating that is right on the horizon. So thank you for bringing that topic up.
Thank you.
Well, Sage, Jimmy Valvano dreamed of a victory over cancer, and what you've seen today is that with the V Foundation, the scientists, and everyone who supports them, we have the right team.
Yeah.
And we are starting to win. We're on a roll, and not even COVID-19 can stop us. There you go.
Incredible. Incredible. If you had to, I'm going to put you on the spot then.
Uh-oh.
If you had to pick one thing, like our biggest takeaway, in your opinion, from what we have listened to today, what would it be?
Well, I love sports metaphors. The immune system is no longer sitting on the bench. We brought it into the game, and it's starting to score points.
We like scoring points. Thank you so much. I want to thank everybody here for coming, those of you, and for following the mandates and helping us out, and especially for those of you who are watching virtually and sending your questions in. We have so many more. And honestly, I think you can tell if you see the doctors out and about, stop them. A couple minutes, right? We're not going to, no full court press, but stop, because obviously they love what they're doing and to talk about it. And so many people are going through so many things personally. So we so appreciate that. Specifically to, yes, Dr. Finn Butterfield, Dr. Lawrence, Dr. Nelson. You're my new bestie. Did you know that? You're so stuck with me. Like, great, awesome. So thank you again, the 2021 Answer for Cancer Symposium. And as I said earlier, the festivities continue. This is actually a nice little preview of the V Foundation's newest fundraiser, basically. It's going to be held in March, the Sonoma Epicurean. And so outside here at Marketplace, I need all of you and everybody watching in your hotel rooms to come on out. Lots of vintners who will be there for the March celebration. Lots of tasting happening right outside at Marketplace. So enjoy as the celebration continues, and thank you. Thank you for everything you're doing.